I'm very, very happy to introduce Professor Bin Yu. Bin is the Chancellor, Distinguished Professor, and Class of 1936 Second Chair in Statistics, EECS, and Computational Biology at the University of California, Berkeley. She's a scientific advisor at the Simons Institute for the Theory of Computing, and her research focuses on the practice and theory of statistical machine learning, veridical data science, and solving interdisciplinary data science problems in neuroscience, genomics, and precision medicine. She and her team have developed algorithms such as iterative random forest, stability-driven nonlinear matrix factorization, and adaptive wavelet distillation for deep learning models. She was a Guggenheim Fellow in 2006, a Tukey Lecturer of the Bernoulli Society in 2011, the IMS Reeds Lecturer in 2016, and the COPS Scott L. Awardee in 2018. At the 2023 GSM, she delivered both the COPS DAAL Lecture, formerly the Fisher Award and Lecture, and the IMS Wall Lecture. She served on the inaugural scientific committee of the UK Turing Institute for Data Science and AI, as well as serves on the editorial board of the Proceedings of the National Academy of Sciences. She's a member of the National Academy of Sciences and the American Academy of Arts and Sciences, and holds an honorary doctorate from the University of Lausanne. So please join me in welcoming Professor Bin Yu.
Well, thank you, Rahul, for the very generous introduction, and also thank you for having me. I'm definitely a newcomer to this community, and I'm really a generalist. I try to combine ideas from different disciplines and hope to develop general principles for data science and machine learning and statistics. So, so in case you're wondering what veridical means, I didn't either. A friend, Tian Zheng from Columbia, suggested the term while I was giving talks on 3 principles of data science: computabilities, predictability, computability, stability. So veridical means truthful. So in the beginning, I was thinking about we want to seek truth. But now I think veridical can also mean that we're truthful to the process of seeking truth. And the paper with my former student, Kang Kumbir, came out 2020. And it's really a really broad philosophical conceptual framework trying to help us to do responsible data science, or AI and machine learning, with a documentation requirement. The goal is really toward trustworthy data-driven results. So I started on this path for the last 10 years because these 2 papers really caught my attention about 10, 15 years ago, where we have very low replication rate in oncology. And more recently, a group of scientists, actually 73 teams of them, work on the same data without worry about data processing, all of that, and for the same social science hypothesis to see whether immigration have like positive effect on immigration support for social policies. And they got results all over the place. So they call it a hidden universe of uncertainty. This is the same data, and they are qualified. So it's not like cargo, anybody can do it. And similarly, there are papers in even earlier neuroscience, also in ecology, in finance. Scientists are really saying that there's a lot more uncertainty that you guys quantifying for us. So since I've been working on cancer detection, my question is like, if I have 73 teams of cancer researchers work on the same data set, do we get the same results? So this is something I think is pretty pressing for us. And of course, data science is not just modeling, which most of the professional meetings, now this one is great, people cover a really realistic range of problems, is about modeling. But every step of this data science life cycle, humans are engaged. And they make different judgment calls. They also make reasonable judgment calls, not completely crazy judgment calls. They might lead to different results. And I did this exercise in my class 2 years ago. I had each team, 3 teams, each actually had an ER doctor through connection we had, and they cleaned the same traumatic brain injury data, tried to predict it's a supervised learning problem of the, whether the kid should be sent to CT scan or not, because radiation can be harmful for them. And one team happily cleaned away 23%. They got the same instructions, and I told them not to talk to each other. And we replicated the same instruction. My TA said, oh, I could have done that, I could have done that. We end up with 10 different data sets. And the variability, the magnitude, is comparable to bootstrapping one data set. So this is a severe case that we cannot ignore the uncertainty from data cleaning. So there are 2 big elephants we need to deal with. Before I say we remove it, at least we should address them. First, we need to assess whether data cleaning decisions, processing decision will make a huge material difference, and what to do about it. Maybe harmonization is one solution. Maybe you should keep 2 copies of same clean data and run pipeline if you cannot agree on some harmonization. Harmonization should be forced. I think it's only when people actually agree. And the other thing is the team effect, right? When you realize there's a team, do you do standardization methodology, or you leave it there? Maybe the 2 approaches both viable for this moment in science, and we keep both of them. So I'm saying, before I say remove them, now I'm like retreating from that. We should address them, and at least make it transparent. And so because of this framework, we developed actually through a cellular biology problem, derived biology, into a software line, also a neuroscience problem. And then we later tried to apply the same principles, and I got engaged with the early cancer detection network of National Health Institute. So prostate cancer for this group, I don't think I need any introduction. And the rate of AUC curve is 60% for the man who already designated under the current procedures for biopsies. And biopsies for prostate cancer are actually quite invasive and costly. So if we can reduce the false positive, the question is what's the cost of maybe we'll miss 1 or 2. That's a medical, ethical problem. I think need a committee to decide, but at least there's need, hopefully, to reduce. And can we do better for this group of men? Another project I've been involved with has been 6 years. It's Charles Zuckerberg BioHub Interdiscipline Award, or Intercampus Award. And many people involved, the people with the red, I should point out my main collaborators. Qianru at Stanford postdoc, my former student at Notre Dame now, and Ewan Ashley, cardiologist from Stanford. These are my main collaborators. There are many, many people engaged. I won't have time to mention all of them. So this problem is try to find genetic components or drivers of this HCM, which is hypertrophic cardiomyopathy, and the left ventricle of the heart is enlarged, thickened, and therefore cause a lot of problem about pumping, and also you have small volume in the chamber, so you have a bunch of problem. And 1 in 500 people in the US have this condition. And so both are data science problem, at least my team's contribution mostly data science, but we really engage, especially with Ewan's team, as a really integrated team, also analyze the experiment result. So I want to emphasize that we should always worry about the future. So the test set for machine learning should be the best proxy for the future. And that will make it clear that when you have data from different hospitals, you don't randomly split them. You put different hospitals as the test and when you train machine learning model. Here, we basically have external validation. It's a different study. And for the second project, it's downstream gene silencing experiments. So it's different in nature. We become a recommendation system. I think a lot of the roles machine learning and statistical model play is actually, can we have higher yields for the downstream experiment, clinical trial, or gene silencing experiments? But we always need external validation. Uncertainty quantification, return to my 2 elephant slide, it's really very crucial for building trust. Statistics, traditionally, we talk about you have a very, very good stochastic generative model. And we do take care of, sometimes not the best way, but we have certain way, we have decades dealing with that. But that's only one source of uncertainty. And now I have told you 2 more. One is from data cleaning. The other is from humans like us make different choices. There are many, many other human choices I'm not formally addressing. But for example, when you do data visualization, I know recently violin plot has become very popular. I like box plot. Do you get the same conclusion in terms of exploratory data analysis? So every step of the way, we should really contemplate multiple ways of visualizing data and to see whether you see the same thing. If you don't, then you need to go to the next level, which one is more trustworthy. So these considerations and reality check, P, I started with P for prediction, kind of reality check, but it's been generalized or expanded to mean reality check, because sometimes maybe you cannot do prediction. Instead of changing the name of P, I'm changing the interpretation. So For the rest of the talk, I want to talk a little bit more about the PCS framework, and documentation is a very big part of the framework, and go back to prostate cancer and HCM problems, and then report some current work still ongoing, going from stability analysis is really like qualitative weighting uncertainty quantification for more precise coverage type of problem and compare with conformal inference. So I said, this is really try to make one culture. If you know Leo Raman's paper, two cultures, machine learning versus generative statistics, this is try to marry them and make into one. And in one slide, it's a synthesis. If a lot of you say, I'm doing that already, that's wonderful. It's not saying that everything's new here, but the synthesis, branded, give a name, I think make it standardization easier to compare, and hopefully more people will use it. And it's also expansion. I haven't seen anybody formally taking into account data uncertainty in the quantification process, and hope to build a platform for everybody to engage. And again, in a nutshell, the P now is really reality check, right? You probably should not just look at average prediction profile. You want to look at subgroups. You maybe look at even individual level. And S is really shaking. It's a common sense principle. There's a very good quote from Plato saying that knowledge has been tied down, unlike your opinions, which changes from day to day. It's really trying to say, oh, these different nodes you can change for the whole process. You put a different part, would you have the same result? Of course, this process is not just cycle this. When you do these cycles, from here, you go back to problem formulation. So there are many, many links I'm not drawing. It's a very nonlinear process. And we have to try to be transparent with that. Let me expand it a little bit on stability. Stability is really expansion on sample-to-sample variability. Try to cover things like numerical stability. And it's really, if you have a perturbation which is reasonable to the process, and you write a document about it, and you assess it, hopefully with multiple metrics. It's not just one metric. So we have a book coming up which deals with at least two formulae from data cleaning and also from model choices. And I think stability is a prerequisite for interpretation. If things are not even stable, if randomly starting point for deep learning give you different importance measures, you don't want to interpret it. Unless you're in development, maybe you want to interpret to make better models. And after you have this prediction check models, and you want to look at aggregation over different versions of it, there are many ways you can do the aggregation. You can take a union in the beginning stage. You want to keep all the options open. Later, you might take an intersection. You might want to take importance, do stable ranking. That's what you will see in the prostate cancer case. So there are many, many ways, and you write a document in your context to make a case this is the right way to aggregate the information. So I was listening to Marinka's talk this morning and thinking that how do I do perturbation for her pipeline? So hope we'll have a discussion afterwards. It's a lot more complex than the problem I have to work with. But I hope I'll convince her that there's still some steps maybe she would want to perturb. Not in terms of wet lab experiment, in terms of this dry lab experiment, that there's certain steps worth probing and change things to see how much the impact is. And even for the problem relatively small I work with, you still don't have time to do every perturbation. You never go home. So again, I'm not removing the human judgment calls. I'm really pushing to the next level. You still have to make meta human judgment calls, and you document about it. That's for yourself and for the people who read your paper to establish transparency and trust. I think most people, if you're transparent, people are more likely to trust you, and you're more likely to trust yourself too. So that's just really pushing for transparency and do perturbations. And this work is really related to earlier work. People call multiverse and forking. So the difference is that this multiverse work usually deal with only the modeling stage. We are like the whole pipeline. And also, they usually say do all possibilities. I think computation is not feasible. So with documentation, I'm advocating that you can cut out some of the possibilities, also do marginal perturbations. So it's a more holistic version of multiverse in the modeling stage. Documentation, documentation, documentation. I don't think there's any easy way out. As many people here will agree, there's no panacea. It's really just good habit and documentation and then debate so that we can get closer to truth. And we have a documentation template at my website. You're welcome to borrow and revise for your problem. So people have asked me, like, the P, C, and S, they're not on top of each other, but they do interact. In some sense, predictability is a form of stability from the training set to the test set. So they are interconnected. And when I started supervised learning, prediction always preceded with stability. But now I see cases that you might sometimes want to seek stability first to reduce the possibility, and then look for a reality check. So the order might change. And I'm writing a paper with a colleague, Haiman Allah, saying, like, I said there's a problem formulation. For generative AI, the problem formulation is not clear. So how to expand these principles that will still be relevant to large language model development? So that's work in progress. So now let me go back to the prostate cancer problem. So we started working with Chen Yan's lab from Michigan because I want to get more people from the early cancer detection network PIs to use that framework. And then I already had a good case study from Emily Anderson on pancreatic cancer use metabolites, and I wanted another case study. So I was introduced to Chen Yan by Zifeng Ding from Hutchinson Cancer Center. So there's two people from my lab, Tiffany at Notre Dame, and now at UC Irvine. And then Yuping Zhang, who is the data analyst. So they already had a paper. They were gracious enough for us to stress test, redo the analysis, what they already put on the archive. So the data was, again, said is a man set for biopsies already. So these people would have, they still did biopsy. That's why we have the labels. And already they end up with other work, 54 genes with clinical variables. They were predicting primary outcome of basically more severe prostate cancer. They already have validation cohort set, ready. So they end up using logistic regression. Logistic elastic net, and was able to use 18 genes. So PSA used basically one KLK3 gene. So they find 17 genes and put it back the gene which already in clinical use. And they got really impressive improvement, again, for the man who set for biopsy, right? So all of this is for the people who probably more likely to have cancer, not the general population. And what you can see that they were able to, for this group, increase the AUC from 60% to 80%. So it's a huge bump. They had an earlier measure called my prostate score, which is bumped up 7%. And there's another test in use, it's 15%. So this is where they were in the paper. So we joined, and we talked to Yuping and saying that what other human judgment call you could have made in the pre-processing of this data is like 700, 800, so it's definitely not the same scale as many other problems. And we did 3 other processing pipelines with her support, saying that, yeah, I could have done that. And then we followed the PCS framework and really look at the performances and compare the different models. And then took the big ones, the good ones, and then did a stable ranking. So if you look at this, what you have on this side, this is logistic elastic net. This is 4 different pre-processing patterns. I don't have time to give you detail, but that's with the support of the person who processed data. So pretty consistent. And this is the second method for pre-processing method, so on and so forth. And we have a new method from my group called FIX. It's the additive decision trees, try to deal with decision trees don't work well for additive models. And this one definitely just don't work as well. So that didn't pass our prediction test. That's a basic meta-human judgment call. And then this is kind of XGBoost. Don't have really good importance measures, so we couldn't use it because we want to rank the different genes. So we took all the other ones and then did a stable ranking. So we have now 5 models. Each model has their own way of importance, right? MDI is what comes random forest, and linear model, you use coefficients. But they still have ranking. So at the ranking level, they can be integrated. Not the p-value because they mean different things. And then we took the average of the good models, or the ones with important measures. And then we just look at plus clinical variables. We look at the first gene, clinical variables, and so on and so forth, until in the validation set, we see something quite similar as the 18 gene. That's where I was stopped. Okay, so it's the ranking that using the multiple methods allowed us to do that. Any questions? Because this is where last time I talked, people have questions. So it's pretty straightforward. So we're able to reduce from 18 genes to 8 genes using multiple methods. And they tried with one method. My intuition is that when you use one method, you have particular way the methodology interact with the dependence structure. Usually, if everything is independent, then you don't have a problem ranking them. The correlation won't interfere. But when you have different structured models, the structure interact with the dependent in different ways. So if some gene come up ahead, can sustain, pass all these different model structures in a way it's more trustworthy. It's closer to causality in a way. And there's also invariance principle in causality. So this is kind of creating a pseudo environment by using different structure models and to allow the screening so that the real important ones come up. So then we went to external validation, right? I was pretty, because I have a lot more experience, but Aru was still waiting for the validation result before he believed what these 8 genes are real. And we did. We lose a bit, 1% to 2%, which is the case. But remember, for data cleaning, we already have 1% to 2% variability. So this is within that error. That's why we saw it's on par with the original 18 genes. And we call simplified MPS. A paper is being revised. And the declaration of conflict of interest, so Aru actually is heading, is part of a startup, try to make this into clinical use, and they're filing a new patent with us as the co-inventors for this simplified, because more likely FDA will approve a panel with 8 genes and 18 genes. My friends in Australia tell me that their lawyer wants 5 genes. So definitely the smaller the better, but the question is whether we might miss some future heterogeneous mechanisms, but that's remained to be seen. So we need to do some enrichment analysis to understand it better. But so far, our collaborators seem to be happy. And that was an unexpected improvement, because we really went in to try to stress test. And because of the stability principle, we actually also gained simplicity. So case study 2 is a lot longer process, a lot more work than what I showed you. I felt like we got lucky with the prostate cancer. But this one was 6 years plus work, and we're still revising the paper. Recall that's the condition, and we use UK bank data, and we're really going for causality here. There was just prediction. That was good enough. I think causality is really a whole spectrum of evidence. It's not about yes or no. If you look at the physics law, Hooke's law, right? It's like pretty stable, mostly replicable. And then you look at the statistical causality. It's really average effect, even when now doing heterogeneous treatment effect. Try to go to small group, which is more relevant to precision medicine. So our premise is that predictability follows for causality. If you add stability structurally and all of that, you get better recommendation for downstream experiments, for higher yields. We don't claim causality, but we want to be better recommenders. So here's a comparison with traditional causal inference with observational data. Usually, there people don't do model checking, which I have a problem with. And we basically use prediction screening or other means, insist on some reality check. And the causality evidence comes in this traditional framework through domain knowledge, context, and you write about it. We explicitly perturb the model and the data to have evidence to find the more trustworthy importance factors, and we want experiment to follow up. And here people usually just claim causality. So this is just a comparison. So this problem, I think maybe I dare to say that maybe representative of a lot of problems we're facing to deal with complex human diseases, cancer or HCM, in the sense that data analysis is not enough. So for us, after many rounds of changing the phenotype and doing many, many things, and stability, all of that, we only got prediction accuracy 55%. Really, really low. But the ranking, using this iterative random forest to do model selection with stability, we identified 3 genes, 2 pairs of epistasis interactions. And we did experiments, or the collaborative experiments. So I think this process might become typical for us to deal with complex human diseases, especially when we deal with epistasis. I think GWAS pretty much did pretty well with the marginally important genes, but the signal is much, much weaker for epistasis. So data analysis, I don't think is enough. You need to combine with other evidence seeking. So this is just a very quick run. I won't have to go to detail. So the methodology is called low signal iterative random forest. Basically, we have to add more stability into the iterative random folder, which was from 2018, to find the gene drivers. And we did a nanotech database search. We did gene silence experiment. And we spent another 8 months trying to use other data to get the mechanism interpretation. So this is a huge effort. So to summarize, we did 5 such experiments. 4 of them yielded very strong causal evidence. We find genes that are possible drivers of this condition, and we find 2 pairs of interactions. So Titin is a well-known heart muscle gene, and CCDC141. The functionality is pretty unclear, but very close on the chromosome. People assumed that they just work together. And what we discovered that they interact, they don't always work together. And this is a growth factor gene. So these 2 interactions, we think, are real. And that's what the paper about. And we're still revising for Nature cardiovascular research. And here's a shout out for my main collaborators again. It was really much a team effort. And we really engaged in the experiment analysis and the redesign experiment. So it's a really integrated team working together with UN's team. So last 2 minutes, I will say a little bit about uncertainty quantification, is that this will try to get a little more quantitative instead of stability, kind of magnitude, kind of holistic, qualitative uncertainty quantification. So chapter 13 of my book, I will mention later, is my former student, Veridical Butter, and then there's the current team members. And this is really trying to add 2 sources of uncertainty beyond the sample-to-sample variability we try to deal with for data collection process. The idea is very similar to Bootstrap. Effectively, Bootstrap, you can have all the asymptotic results you have, but effectively, you're comparing your uncertainty with a fixed set of Bootstrap samples. That's what it is. So in that spirit, we basically create more pseudo data set by considering data cleaning uncertainties and model choices, and use that discrete pseudo data set, pseudo values to assess uncertainty. And we compare with conformal inference because that's something which has been quite popular, both in statistics and machine learning. So PCS is a whole pipeline. This is more on the data side, and they use 1 model, and they also use constant adjustment. We both have a calibration stage, and we do multiple calibration. So high level, we try some data sets without worrying about data cleaning. And just 1 slide to show you that we're still doing more. On the data by relatively low dimension, we reduce the width by 30 to 50%. And for higher dimension, we don't have as much reduction, but still 5 to 15%. The interesting discovery is that we actually have automatic coverage adaptivity to subgroups. In hindsight, it's the reason that when you have lots of data, a lot of signal, all the good models actually agree. When you do multiplication to make the right coverage, you keep that relevant. And they do like constant kind of calibration, then they cannot adjust. So that's kind of the intuition. And what's the secret source? I was giving a talk at Geneva Farm last year. It's really data science hygiene. We have a lot of doctors. I really very much just saying that you have a serious illness. I want to talk to different doctors. And I want to agree their opinion. That's basically the idea. So PCS is really a broad framework. It can be used in many different ways. I showed you the more internal validity. And also uncertainty quantification, and I mentioned the recommendation for external validation because you're really getting closer to causal factors than otherwise. And we have been also using it, like Ethereum, to have new machine learning algorithms. We have a couple of papers on that, and different groups have already extended to other related areas. To summarize, I think I'm probably running out of time, and I hope you take a look. And we have software, try to help the stability analysis vFlow, and we have an R package, try to encourage multiple simulations, a form of stability. Don't just simulate from one model. And we also have merits, which is the simulation guidelines. And that's the documentation you might be able to borrow. And the book. So the book has appeared online, free, at vdsbook.com, and the print version will appear by MIT Press in October. And it's for people to use. It goes really well with mathematical book. We try not to use too many notations, and it's really available for the domain scientists and different exercises. So, thank you. I guess.